So now a more critical part of the nitrogen cycle is the so-called urea cycle. And I like to think of this as the pathway to get rid of nitrogen. And so think about it this way. If you eat too much protein, you'll have too much nitrogen in your body, and we need to get rid of it. Now, the cell can just pee out, or the body can just pee out amino acids, but then that's a waste of carbons because you're also losing the carbon backbone. And so sometimes we just want to get rid of the nitrogen and use the backbone of the sugar of the amino acids for other purposes like making sugar. And so in this case, we use the urea cycle. And essentially, the final product is urea, which is as concentrated of nitrogen as we can have with the least amount of carbons. So how does that work? So there's two ways that the nitrogen enters the cycle and one way that the nitrogen leaves the cycle. It leaves as urea, um, but it enters as in the form of both ammonia and in the form of amino acids. So both ammonia that is kind of free in the cell that is toxic and we don't want, but also some amino acids that just donate their nitrogen. And we'll talk about each step uh, of this pathway. So let's start with the first part. How does it get the ammonia from, or the nitrogen from ammonia? Well, what will happen is that a bicarbonate reaction will get phosphorylated, and then that phosphorylated bicarbonate molecule can now pick up the nitrogen and form what's called carbamic acid. So essentially, if you're looking at this reaction, besides writing it down for your cheat sheet, you should realize, this reaction is probably unfavorable if it was straight, so we use ATP to charge it. Then afterwards, we want to charge for another addition of nitrogen. And so we phosphorylate this other OH of the molecule. And so we consume another ATP. And then your final product is carbomoyl phosphate. Now, some question is that, can you pick up another nitrogen on this left side? I'm not sure. But I think the most common pathway is that this second O is used for picking up nitrogens from amino acids. But I'm not surprised if it might be able to do so if, let's say, you are really in a highly ammonia toxic environment. So that's the first part, making carbomoyl phosphate or picking up the nitrogen from ammonia. Now, the carbomoyl phosphate will interact with ornithine. So if you have a metaphor for the TCA cycle, in TCA cycle, the first thing that interacts is oxaloacetate. In the Krebs cycle, the first thing that interacts is the ribulose 1,5 bisphosphate. In the urea cycle, it's ornithine. Ornithine is this molecule right here. It is an amino acid, but it's not proteinogenic. So we don't see it in our proteins. Essentially, the two of them will interact like this, such that the ornithine nitrogen will basically replace the phosphate group of carbomoyl phosphate, such that you form citrulline, which now has this kind of urea backbone to it. And so that's how the carbomoyl phosphate enters the pathway, by interacting with ornithine. So that's a condensation reaction, essentially. This might be the first time we're seeing the word ornithine or citrulline, so definitely just write it down your cheat sheet. I know this textbook uses a lot of like stick figures, but van der Heijden will probably draw it as a bunch of CH2s. So hopefully you guys are more comfortable with the drawing or conversion of that when you write it on your cheat sheet. Okay, now the next part is where it gets complicated. Essentially, citrulline and aspartate will fuse together. And so the way that works is that citrulline will get charged with ATP first. And in this case, instead of getting charged with ATP, it gets adenylated. So we find AMP instead. So you form citrulyl AMP. And then now that it's charged with energy as citrulyl AMP, the aspartate can now attack. And so essentially what you're left with is the fusion of citrulline and aspartate called arginosuccinate. What's to remember for this pathway besides the structure is that you consume ATP. And actually, because this is an ATP to AMP reaction, this is often considered 2 ATP because we have 2 phosphates consumed. And so if some textbooks or specifically van der Heijden asks you the cost of this step, you can say because it's an ATP to AMP reaction, that it can be considered as a 2 ATP to ADP reactions. Additionally, you'll see that there's no initial thing that leaves off. Initially, that they combine together as arginosuccinate. So, but overall, hopefully you guys are seeing this pattern now. This is an unfavorable reaction, so we charge it with ATP. Now, then we need to get rid of this bulky carbon. So the whole point of aspartate entering this pathway is aspartate wants to get rid of its nitrogen, but we also want to get rid of this carbon now because we want to return it to the cell. And so the way it works then is that the carbon part of aspartate, which is blue in this section, is going to come off. And when it comes off in the form of fumarate. And so I didn't put the arrow pushing here because I don't think it's that important, but essentially we kick out this group right here and we're left with fumarate. And when we're left on the other side, the one with the nitrogen left over is now arginine. And so hopefully this is something that you guys recognize. And then This fumarate will return to the cell. So fumarate can probably enter the TCA cycle and become sugar again. So you can see how aspartate can be recycled into a sugar. Additionally, the arginine can now make urea. So the arginine essentially will kick out this group right here. So basically water will come in to dehydrate this bond in between them. And so what you're left with is the original ornithine and the urea molecule. And so hopefully what you've reached now that we've gone full circle is that we have a circulating ornithine molecule that picks up nitrogen from two origins, from carbomoyl phosphate and from the aspartate. The aspartate can be the influx of nitrogen from excess amino acids, whereas the carbomoyl phosphate can be the influx of nitrogen from just deadly ammonia in the cell. Also, they noted here the locations of where they come from, so maybe that can give you some context. But overall, the thing you should remember from this is that this is our way of getting rid of nitrogen. And to connect this with the gluconeogenesis, the fumarate, like I said earlier, can essentially be recycled to glucose. Similarly, alpha amino acids can give the nitrogen to oxaloacetate, so it becomes aspartate. And then the aspartate will then give up its nitrogen to the urea cycle. And so essentially, this allows you to connect that. Now all these excess nitrogen that we talked about isn't just ammonia. It's also in the form of amino acids, and also this allows us to recycle them. Any questions from the people for real time about the urea cycle? Okay. So with the remaining time, I will try. I realize that there's not enough time to go over the in-depth of nucleic acid breakdown and synthesis, as well as nucleotide. And so I'm going to tell you guys what I think is important, and then you guys can read the PowerPoint on your own because they have all the pathways anyways. And so for the amino acid breakdown, the one thing to note is that we can recycle amino acids and their carbon backbone after we've gotten rid of the nitrogen. The sad thing is that not all amino acids can be convertible into one direction. And so there's so-called glucogenic amino acids and so-called ketogenic amino acids, and then so-called both. Essentially, if you're glucogenic, then your backbone can be recycled into glucose. And so I put in here the list of those. If you are ketogenic, then your backbone cannot be broken into glucose, but it can be converted into acetyl-CoA. And so essentially, you can make fat out of that amino acid, but not glucose. And then lastly, there are some that are both. In this case, their backbone can be used to produce both sugar or fat. And so that's a breakdown of that. Essentially, what you're trying to say here is after you get the nitrogen, is there a pathway that exists to go to sugar or to go to fat? And that allows you to divide them into these three. I didn't memorize any of them. I know pyruvate and things like that is kind of really obvious, but I would just make a list like this. That way you're aware. And also, if they give you an amino acid, then you can double check it to your list and say, hi, is this glucogenic or is this ketogenic? Am I, is it impossible to make this pathway? And so on. So all of the reactions that are going to be shown in the amino acid synthesis pathway are going to be things you've seen before. And so hopefully you guys can kind of see a reaction and realize the pattern. If I show you I will upload a better version of this with actual. I'm sorry, I didn't know why the images didn't work well. Maybe it's because it's a PowerPoint, but certain images are retained or not. But essentially, if we look at the pathways on the left here, you'll notice that a lot of them are pathways we've seen before. Deamination, so it involves PLP. Alpha decarboxylation, so it involves the TPP lipoic acid mechanisms we've learned before. Oxidation probably needs NADPH. And then hydrolysis needs water. And so all of these pathways we've seen before. So hopefully you guys can understand that. It's what's more important for you guys on the exam is just to write down the pathway. I know in this recitation at the current moment I don't have them. I will post the more corrected version of this definitely. But I put in here all the amino acids that I think you need to know how to synthesize. So here, glutamate, you can make it from alpha ketoglutarate. Glutamine, you can make it from glutamine or glutamate. And then alanine and aspartate can be made from the nitrogen transfer to oxaloacetate and pyruvate. And then arginine can be made from the urea cycle. Serine can be made from a backbone of 3-phosphoglycerate. Glycine can be made from serine. And I think those are the most important ones. The tyrosine and other ones, those are hard because they're aromatic. And so less focus is given on them. But I think all of my other mechanisms are intact except for these two. So I will make sure to fix that by the time this recitation gets uploaded. But overall, hopefully the pathway is the same. You should notice every pathway is about, you've seen them before. So hopefully they're not super new. The other part is the nucleotide biosynthesis. The only thing strange about this is that there's two ways to make nucleotides, and that is to recycle them, called the salvage pathway, or to make them from new, so-called de novo pathway. Um, and so instead of amino acids just being kind of nitrogen transfers, a lot of times when you make nucleotides, we often like to recycle. So just something to keep in mind. And you guys can look at this and you'll see there's a lot of like acronyms GAR, FGAR, and all of that. It's not super important for you to remember the name. So when I recommend that is if you put it on your cheat sheet, instead of writing glycinamide ribonucleotide, you just write GAR. Additionally, you'll notice that also all of the steps are things you've seen before. The glutamate transferred the nitrogen to become, the glutamine transferred a nitrogen, so it became a glutamate. So things you've seen for PLP. And here you have a THF transferring a formyl group. So in this case, this is a THF mechanism that we talked about with the formate. So all of these pathways you've seen before. The only thing that's probably new to you is that you're making a ring and that you're making molecules that are like much more complicated than amino acids. So what should you focus on? Assuming that you have this on your cheat sheet, so you don't need to memorize it, what you should focus on is maybe color coding them. So if you see this diagram I'm showing right here, I'm showing the production of a purine, but notice how the carbons are colored. And that is done for a reason because it tells you where they come from. And so what's important for you to know is when they give you a final product, where do the carbons come from? Which carbon is from aspartate? Which or nitrogen is from aspartate? Which carbon is from glycine? Which carbon is from THF? And so when you study these later on, make sure you focus on those because that is the most critical part in the labeling part. He'll ask you definitely, I have radio labeled glycine, where would that show up in my nucleotide? So that's 3 points that hopefully you guys won't lose. And I put in here kind of a guide for that, so you guys can also refer to this. Generally, the first product that they make is what's called inosine. So generally, inosine can be made into AMP and GMP. So something to keep in mind where the pathway diverges. And then for pyrimidines, I'll send you guys a more fixed pathway where I'll show you guys that as well. And usually the intermediate that diverges for that is the so-called UMP. But other than that, I think every pathway has kind of mechanisms you've seen before. So hopefully you guys aren't super shocked about it. What's more important for you is to just make sure you have it on your cheat sheet and to color code so that you know where the stuff comes from. Because that is the, when he asks you synthesize cytosine for me, then it's an easy copy paste for you guys. But if he asks you which part of cytosine is from glycine, then instead of having to backtrack the pathway and track where the carbons come from, then you can just kind of have an easy check of where that carbon comes from. And like any other pathways, this is also inhibited by products. So not super important for you to memorize that, oh, this step is super critical, but most important for you to realize is that the things that inhibit this pathway are its own products. So hopefully that is something that you've already learned from taking this entire course. I will upload a more corrected version of this because I think the PDF is not showing the correct version because of the images. So I will try to fix this. But overall, I will say you guys are pretty prepared to kind of just like take this pathway because all the mechanisms you've learned before. But I told you guys what to focus. So hopefully you make sure to remember that. I know I didn't cover a lot about the nucleotides, but for those in real time, do you guys have any general questions? Okay. If not, this is the end of recitations. Thank you for watching them and attending them. I know that this is a weird time because of online teaching, and also it's my first time recording anything similar to online learning, so hopefully you guys are patient for me, so thank you for that. I don't know when student evaluations are out yet or not, so if they are out, please do take the time to let me know and let us TAs and professors know what we can improve on. Especially regarding this online learning. But other than that, if you guys have any questions coming up to the final, you guys can always message the, what do you call this, the Piazza, and also you can all contact us by email. I don't really have a final, so I think you guys can always reach out as well. But yeah, and if you have any questions, just don't hesitate to ask. Other than that, I will hopefully see you guys sometime in the future.
Thank you.